Embryology explores the formation of organisms from fertilization to complex structures and systems. Join me as I delve into the intricate processes that shape our bodies, focusing on the molecular and cellular mechanisms behind the development of the essential pituitary gland. The pituitary gland undergoes complex development regulated by genes like PIX1, PROP1, and LHX, and signaling molecules like SHH, FGFs, and BMPs. These factors determine cell fate and enable proper pituitary gland formation. Transcription factors are key regulators in the process of cellular differentiation, playing a crucial role in pituitary gland development. Through their interactions with DNA, transcription factors such as PIX2, PU1F1, and PROP1 determine the fate and identity of specific pituitary cell types, including somatotrophs, lactotrophs, thyrotrophs, and corticotrophs. These factors activate a cascade of gene expression events, leading to the acquisition of cell-specific characteristics and the secretion of particular hormones. A complex network of signaling pathways is responsible for guiding pituitary morphogenesis. Among these signaling pathways, the Notch, WNT, and FGF pathways are of particular importance. Notch signaling regulates cell fate determination and proliferation, while WNT signaling controls cell polarity and tissue organization. FGF signaling, on the other hand, influences cell survival, migration, and differentiation. Crosstalk between these signaling pathways ensures proper proliferation, migration, and differentiation of pituitary progenitor cells, ultimately leading to the establishment of a functional pituitary gland. During the formation of Rathke's pouch, a precursor structure for the anterior pituitary gland, several complex cellular events occur. These include invagination, expansion, and remodeling. Signaling molecules like BMPs and FGFs and transcription factors like OTX2 and LHX3 are instrumental in this process. In the video on the development of the pituitary gland, we will explore the complex interactions between the gland and neighboring structures. Molecular cues from the oral cavity and brain play crucial roles, while the gland itself influences the development of these structures through hormone secretion. This reciprocal influence ensures the proper coordination of endocrine and neural processes. The anterior pituitary gland is composed of different cell types that release specific hormones. Understanding how these cells differentiate and function is crucial for understanding hormone regulation and its effects on the body. Stem cells are important for maintaining and potentially regenerating the pituitary gland. Specialized pituitary stem cells exist in the adult gland, renewing damaged or aging cells. Studying how these cells are regulated is essential for improving pituitary function and repair. Epigenetic modifications play a vital role in establishing and maintaining cell-specific gene expression patterns during pituitary gland development. DNA methylation, histone modifications, and chromatin remodeling complexes act as regulatory mechanisms that determine whether genes are activated or repressed during cellular differentiation. These modifications influence the accessibility of genes to transcription factors, ensuring precise gene expression patterns. Disruptions in the epigenetic landscape can lead to developmental abnormalities and diseases associated with pituitary gland dysfunction. The development of the pituitary gland can be affected by both genetic and environmental factors. Genetic mutations in critical genes involved in pituitary gland development can lead to various congenital abnormalities, such as pituitary hypoplasia or aplasia. Additionally, environmental factors, including exposure to toxins, maternal hormone imbalances, and certain medications, can disrupt normal pituitary gland development. Understanding the interplay between genetic and environmental factors is essential for diagnosing and managing pituitary developmental abnormalities. The development of the pituitary gland exhibits species-specific characteristics and evolutionary adaptations. Comparative studies across different mammalian species, including rodents, primates, and marine mammals, have revealed variations in the timing, morphogenesis, and functionality of the pituitary gland. By examining these differences, we gain insights into the evolutionary history and adaptive significance of the pituitary gland, ultimately enhancing our understanding of its development in humans. Connective tissue and vasculature play essential roles in providing structural support and facilitating hormone secretion within the pituitary gland. Developmentally, a complex network of blood vessels is established to supply oxygen and nutrients to the gland. The precise formation and maturation of this vascular bed, along with the structuring of extracellular matrices, ensure proper functionality of the different pituitary cell types. Perturbations in connective tissue development or vascularization can lead to vascular malformations and impact hormone secretion. The neurohypophysis, also known as the posterior pituitary gland, arises from a distinct lineage and exhibits unique developmental processes. Neural precursor cells originating from the hypothalamus migrate along specialized cellular tracts to the site of posterior pituitary formation. During this journey, these precursor cells establish neural connections with the hypothalamus and differentiate into neurosecretory cells, which secrete oxytocin and vasopressin. Elucidating the molecular and cellular mechanisms underlying neurohypophysis formation provides critical insights into the integration of neural and endocrine processes. The development of the pituitary gland requires intricate molecular, cellular, and genetic processes. Understanding the genes, signaling pathways, and transcription factors involved, as well as environmental and epigenetic factors, helps us study and treat pituitary abnormalities and diseases. As medical students, you now have a comprehensive understanding of pituitary gland embryology and its implications.